drug substance. Now, why I go into this is because these early discovery under the fixing of and locking of a process to make a bulk or as a final vial is very important for clinical trial to enable this, but it has also prospective importance because it is the consistency of the product that is necessary, because all the testimony that will be supplied in the submission of efficacy and safety depends on the product that is actually undergone examination in clinical trials. Now, if then the clinical trials have finished, then of course you have to swear back that they were, what it then produced is exactly has these properties. So the efficiency and the product safety is still the paradigm of biotechnology, is still the product is the process. And there are multiple process limitations for process development after clinical testing to maintain a full product, but these all can be done, but they need a very stringent comparability test. And here is an elaboration on the same iceberg that we saw before, where it says, okay, this is what we can see with direct testing the final product. Then there is something that we do not test on every product, and that's the characterization of the product that the process does. Then the process has a number of features that are tested in process control testing, but it is also rather important that what happens to raw materials that go into the process, procedures of monitoring, and the validation. And beyond that, there is still an open thing, an unknown, which sometimes comes only out when you have run a number of clinical tests and see something there. So this is why the release of a product, albeit just with a certificate of analysis, it is based on the foundation of enough of knowledge that really makes it certain that this product is as has been tested in the clinics. This tie is very important because this tie has created a lot of, people would like to say, inertia in innovation of manufacturing of pharmaceutical drugs. So, now these processes will be applied to pharmaceutical proteins. The proteins, however, they have their own thing. They are not all needed in the same amounts. They are also need therefore in different operational and scales, and they require whatever is available or can be made available as a manufacturing site. For demonstration, we've had all these numbers. Here you have a number of successful biotechs from Avonex to Malthera rituximab, and this is ordered in the sequence of the dose that is usually applied, or if you want from a manufacturing point of view, in what is the amount that's required for a patient treatment per year. And you can see it goes from milligrams per year to grams or kilograms per year. And so it is very obvious from this that not every product requires the same kind or the same size of a facility. So the output of a biotech plant is then to install the installation hardware has to be run between a minimum and a maximum to adjust to the market volume. So each product has a specific scale. As I said before, the amount is dose, treatment, and patient number, and the supply that needs to match these is productivity, inventory, and a number of strategies, including the hardware of the available manufacturing. So we have very different sizes of facilities for every protein and protein candidate. The normal standard platform that has evolved so far is usually the one that I have depicted before, and it starts with the a basic growing vessels and then a downstream area that then handles whatever comes out from here. And here is just an attempt to see how large the vessels need to be in order to do that. So the manufacturing process for master cell, then seed train harvest is down here, and then the purification to drug product. Here is an attempt to show these are the volumes of vessels that needed to be there. And you can clearly see that in the early days with a traditional titer, the largest volume has always been the bioreactor in which the product was made or harvested. And from then on, in particular for antibodies, it went down very much into protein A, and then sometimes it went up again because of columns to have a negative or bind a dilute or flow through columns. With very low titer, the volume may still be the same, but then you get this on a different method, and the amount of vials in the very end is then dependent on the activity of the dose per patient. So the facility design for recombinant proteins can therefore evolve from manufacturing process for large scale in a facility first as a mono-product operation. That has been an adoption from chemical thinking that you have a facility that is usually dedicated to one chemical. This is clearly then being challenged because of the variation of need as well as the variation of titers that could be reached, and therefore very quickly there was a possibility that the FDA was actually able to adopt, and that was a multi-product plant. So that we had multi-product plant, so even with large scales and six-pack, 6 bioreactors as the largest scale, became the most prominent large-scale manufacturing facilities for proteins, in particular also for proteins of monoclonals. And these were then run. This is the setup that is there. So you have 6 bioreactors, 6 cylinders, and they have a number of seed trains, and they have 1 or 2 downstream areas. And you can run all of these in parallel or together, depending. It creates an enormous flexibility that is necessary for good occupancy of the plant and therefore for good economics. To build such a plant, it takes about 7 to 9 years in a green field to get it from feasibility studies to approval. This can be accelerated depending on what you have, but the green field is, this is the example here, is perhaps the longest. And it is something that takes a lot of upfront money and investment, just like Kat mentioned before. A plant of this order is in the order of $300 to $500 million. This is the plant in construction that Biogen has built in Research Triangle Park. The large-scale facility is this one. You can see that it is still here in construction. When it works, this is the plant as it exists today. And we have the large building here houses the large column and the cell culture and bioreactors. We then had as a second floor, a second purification site, purification suite. The warehousing and the tank farm are here, and in this building is quality control. Inside of such a plant, you can see bioreactors. These ones are 200-liter bioreactors, and they go larger. You cannot see the 15,000 liter bioreactors here. These are the heads of them only, because they go through several. They go through several stories. The largest vessel in the building is this one, which is a 25,000 liter harvest vessel, which is the feedstock for the downstream purification. Then this is what we call flippantly our Our organ in this are tangential flow filtration and hollow fiber format to separate cells from the supernatant, or this one here is a bowl centrifuge, continuous centrifuge, in order to do this by centrifugation. In the end, these are the columns. This is the 160-meter column that is used for protein A or ion exchanges to purify in several batches. So this six-pack started here and has been built for many of these products that came along even today. And so we have, after the first, a distribution, worldwide distribution of six-pack facilities that are allocated here from the very first with Genentech here and the Boehringer Ingelheim here. There on the East Coast, there's some in Puerto Rico, there are some in Singapore, there may be some here in Celtron, in Korea, and I'm not so certain what is presently available in Japan. But you can see that this is what the world capacity is today. Given this situation with so much capacity, the question is for everyone how to secure the capacity for supply for anyone who has something new. Do you want to build or buy? What is the life cycle of the drug? What is on the market available as commercial? And what are domestic regulations conditions to make it possible in the country where you want to do this? And here, just two slides for what the consideration of a manufacturer is. You have these large plants, and in these large plants there is